How's it going, you guys? So in this video, I wanted to discuss something that I've seen show up on past AP exams, and I've seen AP exam questions that deal with this, and that's action potentials. Now, action potentials have to do with neurons, and neurons make up our nervous system, so it allows us to feel things, allows us to think things, and ultimately neurons are these cells that conduct electrical impulses throughout our body. So an action potential is basically looking at a single section of the neuron and seeing what's going on to allow that signal to be passed down from, let's say, my hand to my brain when I touch something. So first off, I want to discuss a little bit of neuron anatomy before we get into action potentials. So again, neurons help us feel things, they help us conduct electrical signals throughout our bodies. They're among the largest cells in our bodies, and they have a couple parts that you should know. For starters, the central part that has the nucleus, that is called the cell body. The cell body. These extensions from the cell body towards the top, those are called dendrites. They are cellular processes that receive signals from other neurons, so dendrites. This long process right here is the axon. And the ending points of the axon are the axon terminals. Axon terminals. So those are the parts of the neuron that I feel like you should know before we jump into this. And generally speaking, the pathway of electrical conduction passes from the dendrites to the axon terminal. So this is where the signal is received. I'll put signal. Signal is received. And then at the end, at the axon terminals, this is where the signal is transmitted. And it's transmitted onto the next cell. So those are some main parts of the neuron. That's the neuron anatomy, if you will. So now that we know these parts, imagine if we were to zoom in right around here. Right at this part of the membrane of the axon. So let's zoom in there. And we see this. So let me just make sure everything's in frame. Good, I'll move this over just a little bit. All right. So we are now looking at the membrane, which is part of the axon. So we're looking at a zoomed in version of the axon. So out here, this is outside of the axon, so I'll put outside of cell. Outside of cell. And in here, this is the cytosol, so I'll put cytosol, which is inside of the cell. So now that we have our bearings, outside of the cell, inside of the cell, here is our phospholipid bilayer that we find in any cell membrane. And if you notice, there are a couple of proteins embedded in the membrane. Here we have a mechanically gated channel. Now when we say gated, that means it opens. And if it's mechanically gated, that means it's opening through some sort of physical stimulus, like maybe perhaps pressure. And here we have a couple of voltage gated channels. They will open when the voltage changes. So when the relative charge between the inside and the outside of the membrane changes, that's going to stimulate these ones to open. And we'll talk about that in a second. And then this is a protein pump. This is a pump that you might be familiar with, the sodium-potassium pump. It needs energy to work, but it will ultimately move ions against their concentration gradient and kind of restore the concentration gradient that I'll talk about in a second. And don't worry about this voltmeter just yet. We'll get to that in a second. Also notice we have some sodium on the outside, and we have some potassium ions, so sodium ions and potassium ions on the inside. Now, one thing that I want to mention is right now we are looking at a resting membrane, and therefore its membrane potential, its relative charge between the inside and outside of the cell, is at the resting potential of negative 70 millivolts, roughly. So we right now have more sodium ions outside than we have potassium ions inside. And we also have some proteins that are just kind of floating around in the cytosol. They typically carry a negative charge. So all in all, the inside of the membrane is more negative. So the cytosol is more negative than the outside of the cell, the extracellular matrix. So the cytosol is more negative than the outside of the cell currently. Now, we are at resting state, so nothing is happening. Now, I want you to imagine if I were to touch this whiteboard, there is going to be an initial stimulus, perhaps an initial pressure, and that pressure is going to cause a change at the cellular level. So let's say there is some amount of pressure put on this membrane. This membrane might open up. Now granted, this part right here, this mechanically gated part, only happens in the initial neuron. It wouldn't necessarily happen on the axon. It'll happen at a receptor. But some initial stimulus causes one of these mechanically gated channels to open. So this is a mechanically gated sodium ion Na+ channel. Now what's going to happen is this initial stimulus is going to cause this channel to open. Now when it opens, you might have noticed that there is a higher concentration of sodium outside of the cell than inside. So what's going to happen is as soon as that opens, sodium is going to rush into the cell. So sodium will rush into the cell. Now what's that going to do to our membrane potential? It's going to change it. So we are going to have more positives moving inward, so that's going to make the inside more positive than it was before, and the outside, since it's losing positive charges, it's going to be relatively negative in comparison. So what's going to happen is this membrane potential is going to go from being negative to more positive, because we're seeing more positive enter the cell. And as that does that, if it reaches a certain threshold, if there's enough sodium that rushes in, it's going to trigger this voltage-gated channel. So I'm going to do a little, maybe like a lightning bolt. Like boom, boom, boom. So this right here is a voltage-gated channel. Once we hit a certain voltage, a certain change in voltage, this is going to open. Now this one is a voltage-gated sodium channel. So a voltage-gated sodium channel. So an initial stimulus caused this to open. So let me just draw this open, just so we can see this a little bit better. So now, this is open. Kind of a bad drawing, but it's okay. So now that this is open and sodium is rushing in, if enough sodium rushes in, this one will open. So let me have this one opened up a little bit. So this will open. And that will cause more sodium to rush in. Now, as long as this initial stimulus is going, this mechanically gated channel will stay open. If it's not, once the stimulus has been removed, then this mechanically gated channel is going to close. Now, this voltage-gated channel is going to open. It's going to let sodium in. So, as you might expect, if we let even more sodium in, the inside of the membrane or the inside of the cell is going to become more positive. So as it grows in positivity, that will trigger the next voltage-gated channel, and then the next, and then the next. So So all of these sodium channels, it's basically like a domino effect. All of these sodium voltage-gated channels are opening down the road, and that's going to cause a change in the membrane potential and an overall change in charge, which will carry an electrical signal. Now, it's going to rise. So the charge inside of here is going to rise to a certain positive level. Now, at a certain positive level, this right here, which is a voltage-gated potassium, K+, channel. So this one's a little more rigid in its way. It doesn't change as soon as the voltage changes a little bit. So if the voltage changes a little bit, this is going to open. But if it changes a lot, so now we've let in a lot of sodium, that is going to change the net charge to, let's say, positive 30. Before we were at negative 70, now we're at positive 30. So once enough sodium has rushed in, if there's a lot of it in there, what's going to happen is two things. One, this little domain, this protein domain, is going to swing in and block the sodium channel. So it's going to prevent any more sodium from getting in. So we let a lot in, and eventually it's going to block, and this potassium channel is going to open. So this potassium channel is going to open, and now we have more potassium inside of the cell than outside. So potassium will naturally move down its concentration gradient, and we'll get potassium outside of the cell. So just to kind of recap what we've done so far, the mechanically gated sodium channel opened because of some sort of stimulus, maybe a stretching of the membrane because of pressure. And then once that opened and let a certain amount of sodium in, this voltage-gated channel opened and let more sodium in. And then that will trigger the next channel to open, and the next, like a domino effect. But at a certain charge, we need to kind of reset the membrane. So at a certain charge, this will be blocked. So we'll block the sodium channel. We will open the potassium channel. So now instead of sodium rushing in, potassium rushes out. So now we have positive charges moving out, and that will kind of restabilize the membrane. And so this right here is repolarization, where before it was depolarization, now we're repolarizing the membrane. And that's going to kind of bring us back to our negative 70-ish range. And then this last protein, which is a sodium potassium pump. This last protein is going to reset the membrane. So we could hypothetically send another signal. So what this protein is going to do is it is going to act like a carrier protein, and it's going to grab that potassium that moved out and bring it back in, and it's going to grab that sodium that was inside and pump it back out. So it's acting as a pump that is bringing potassium back in and sodium back out to restabilize our membrane. So I know that was a lot to think about, and also this voltmeter is there just to measure the changes in voltage. And so we could actually use a voltmeter if it's small enough to figure out membrane voltage changes, and we can plot that on a graph, which I'll show you in a second. So just as a recap, mechanically gated sodium channel opens, lets some sodium in. Voltage gated sodium channel opens, lets more sodium in. Once we hit a certain point, and also once sodium comes in, that will open up the next voltage-gated channel, sodium channel, and let more sodium in. That will open up the next voltage-gated channel and let more sodium in. And then at a certain point, this sodium channel will get blocked, and this potassium channel will open. So we let sodium in, now we're letting potassium out, which is positive. So we're resetting the membrane, at least in terms of charge. And then we finally, once we reach a certain point, all of the channels will close again, and then our sodium-potassium pump will just restore our membrane potential. It will pump sodium out and potassium in. So hopefully that made sense. If you're still a little bit confused, I do have an animation. Sometimes it's better to see this animated. So let me move over to the animation. All right. So here we have our different parts of the membrane. Notice we have a lot of potassium on the inside and sodium on the outside. The general net charge is positive on the outside because we have a lot of sodium. And even though we have positive potassium on the inside here, we also have some negative charges like our negative proteins. So the net charge in comparison to the outside is negative on the inside. So if we were to go through this animation, the first thing that's going to happen is some initial stimulus that is going to cause the membrane to stretch, and this mechanically gated sodium channel is going to open. Once that opens, it's a sodium channel, so it's going to let a lot of sodium in. Now, if we let enough sodium in, that's going to change the voltage, change the net polarization between these two sides of the membrane, and that's going to trigger this voltage-gated channel to open. Now notice, this channel closed. That's because the initial stimulus is no longer there. So if I brushed up against something, that initial stimulus is going to be brief, but so as long as that stimulus is there, it'll be open. If it's not, it'll close. Now the voltage-gated channel opens. It lets more sodium in. And when that lets more sodium in, that's going to change the voltage on the inside, or the net charge on the inside even more, which is going to trigger the next voltage-gated sodium channel to open. And that will bring in more sodium, and this will be a domino effect. So you can imagine that the signal is going to be passed down or propagated down the axon, down the neuron. Now, if we come back to here, once we have enough sodium in, and we have enough positivity in here, that's going to trigger two things. One, this sodium channel is going to be blocked, and two, this voltage-gated potassium channel is going to open. Now that this channel is open, because we had a higher concentration of potassium in here, that is going to move out towards the extracellular side. And once that moves out, that will kind of restabilize the charges across the membrane. So yes, we had some positive sodium move in, but then we had some positive potassium move out, so now we're restabilized in terms of charge. And once we've reached that charge, all channels will close. Now, again, you might be thinking, and we talked about it earlier, we now have potassium on the outside and sodium on the inside. So if we want to send another signal down the neuron, so if we want to be able to fire another message, we're going to have to reset the membrane. So we have the sodium-potassium pump that, with the help of ATP, cellular energy, it's going to move sodium back out and move potassium back in. So it's going to act like a almost swinging door where it brings sodium out and then pumps sodium or potassium back in. So that is the action potential, and that is the animation of the action potential. So hopefully that made sense. But let me also tie this into something that you might see on the AP exam, and that is an action potential graph. So here is what an action potential graph would look like if we plotted membrane potential on the y-axis and then time on the x-axis. So initially, we have more positive charge outside of the cell and more negative charge on the inside of the cell. So I'm not sure if you can see this. Let me see if I can move you a little bit closer. Here we go. So we have a lot of potassium outside of the cell, sodium outside of the cell, and a lot of potassium on the inside of the cell, but inside is more negative relative to the outside because we have a lot of sodium, and yes, we have some potassium, but we also have those negative protein charges, and so we have less potassium on the inside than we have sodium on the outside. So all in all, inside of the cell is relatively negative. So we say that the membrane potential is at roughly negative 70 millivolts. And so that is resting membrane potential. I'll put resting membrane potential. We also would consider this to be polarized. So we say that it's polarized because we have different sides, right? We have a positive side and we have a relatively negative side. So the membrane is polarized. Now, some initial stimulus causes that mechanically gated channel to open. It could also be a ligand gated channel, which has some sort of chemical bind to it to open it. But some initial stimulus causes that first channel to open. Now, if enough sodium rushes in, and we hit roughly negative 55 millivolts. So enough positive charge comes in, so we're becoming more positive. If we hit that negative 55 threshold, so this is a threshold, we will enter an action potential. Now, if we don't hit that threshold, let's say the mechanically gated or ligand gated channel opened up a little bit, let a little bit of sodium in, but then quickly closed, and it wasn't enough to pass this threshold, it will do something like this and just return back to the standard negative 70 resting potential state. And we call that a graded potential. A graded. So that basically means graded potential. That basically means that we did not have enough of a stimulus to send or fire a response. So that is a graded potential. Now, if we do pass the threshold, so let's say we get enough of a strong enough stimulus, if I get a little bit of wind, I might not feel it, but let's say I get a boulder hit my hand, I would feel that. So we have enough of a stimulus that lets in enough sodium. And now we have the voltage-gated channels opening, the sodium voltage-gated channels opening, letting in more sodium, and that increases the amounts of positive ions entering the cell. So gradually we see our membrane potential increase. So we call this depolarization. So depolarization happens because those sodium channels are opening up, the voltage-gated sodium channels. So they're opening up and they're letting more sodium into the cell. Now, once we get enough sodium into the cell, right here, at about positive 30, now this might change depending on what you're looking at, the source, but we'll say positive 30, maybe positive 40 millivolts, at around that potential, that charge, we will see the sodium channels. So sodium channels get blocked. And these are the voltage-gated sodium channels. So they get blocked. They don't necessarily close, but they are blocked. And we see those potassium channels, the voltage-gated potassium channels, open. And that allows potassium to leave. So now that we're having positive charges leave, we're going to return to our initial membrane potential. So this is called repolarization. So we were becoming depolarized until the sodium channel gets blocked and the potassium channel opens, and then we enter repolarization. Now we're going to continue letting potassium out, and I apologize about this, but we will continue letting potassium out until we reach that negative 70 voltage, and we will go a little bit further. So this is what we call hyperpolarization. hyperpolarization. It's brief, but we have a brief period of hyperpolarization, and then all channels close once we hit that -70. So all channels will be signaled to close, and then we return to our normal state with the assistance of the sodium-potassium pump. So remember, we're pumping potassium back in, we're pumping sodium back out. So I'll put right here, sodium, potassium, pump. And then we return to our resting state, our normal polarized membrane. So this graph might be something that you see on an AP exam. So that's why I wanted to go over it. I wanted to go over action potentials and all that stuff. If you have any questions, feel free to ask me when you see me next. But besides that, hopefully that was helpful. Hopefully that was understandable. And yeah, again, if you have any questions, just let me know.